Hi guys. So our independent research project is going to focus on how effective preservatives like sodium benzoate and citric acid are when used um, by themselves or within food products and killing bacteria like E. coli or salmonella. So as we know, food is required for all living organisms to live. It provides the proper nutrients for energy, growth, and efficiency of bodily functions, and as well as the materials to maintain a healthy immune system. When food becomes contaminated with pathogens like E. coli and salmonella, it can have disastrous effects on the functioning of the human body. Bacteria like salmonella is attributed to an estimate of over 1 million food poisoning cases in the United States, as well as over 200,000 hospitalizations in a year. E. coli is a naturally occurring bacteria in the human biome. When out of balance, it may cause food poisoning. Preservatives like citric acids utilize their acidity to make it harder for bacteria and mold to survive and reproduce. Sodium benzoate is another preservative mostly used in processed foods and beverages. Preservatives help prevent decompositions of microbial growth or unwanted chemical changes in food products.
I'm going to be discussing the different methods that we would have used in lab to go about this experiment, as well as the methods that people used in our research in order to conduct the results for this experiment. So the first method that would have been done would have been using various dilutions of preservatives and a sample of bacteria and determine a growth curve. So in our case, we were going to be using sodium benzoate and citric acid for the preservatives, and then for the bacteria, we were going to be using E. coli and salmonella. So we would have done different dilutions of these and then determine the growth curve, and then whichever curve had the least amount of growth would have then told us that that preservative was more effective at eliminating the growth of the bacteria. So the second method that we would have used would have been plating. So for this method, we would have to have diluted the preservatives, and this is due to them not being in a liquid form. And we would have diluted those into PBS, and then we would plate those dilutions along with bacteria sample onto a plate. And then after incubation, we would have then counted the colony growth on each plate, and then whichever plate had the least amount of growth would have told us that that preservative was more effective at eliminating bacteria. In this specific experiment, we would have used MacConkey agar and TSA agar. MacConkey agar is specifically selective for the E. coli, so we would have used the E. coli and the two preservatives on just the MacConkey agar plates, and then the TSA agar is just selective, is selected specifically for the salmonella, so we would have used the TSA plates to plate the dilutions of the preservatives with the salmonella. And then, so method number 3 would be using Petrifilm. So for this method, we would once again dilute the preservatives in PBS due to them not being in liquid form. And then we would take these dilutions as well as bacteria and then plate them and then put them on the Petrifilm. And so after we'd incubate, we would count the colony growth on each Petrifilm using the Sharpie method that we have done once before in lab. And then whichever film had the least amount of growth would have told us that that preservative on that film was most effective at eliminating the growth of the bacteria. So for this specific experiment, we would have used coliform and Salix Petrifilm, and then this is due to the coliform being selective for E. coli, and then the Salix being selective for the salmonella.
Results. A study was conducted by Dr. Musukia of the International Journal of Food Science that was aimed at researching growth of bacteria using different preservatives. The food product which was used for the study was orange flesh sweet potato puree, which was considered a versatile food product for the purposes of the project. In the non-supplemented purees, which means the purees that didn't have any food preservatives, the E. coli growth increased from 7 log colony forming units per gram to 9 log colony forming units per gram. On the other hand, all other combinations of the puree and the various food preservatives led to a significant decrease in colony units, which is shown on the figure on the left. Similarly, the They also studied Staphylococcus aureus growth in the pureed food product, and they found that sodium benzoate and potassium sulfite preservatives led to a significant decrease in microbial growth compared to all other treatments. Specifically, it led from a 7 colony forming units per gram to 3 colony forming units per gram over the course of the entire study. Furthermore, there have been numerous concerns regarding the harmfulness of food preservatives in the food industry, and a survey conducted by the American Public Health Association on the Department of Agriculture demonstrates that food preservatives, other than usual condimental preservatives such as sugar, salt, spices, alcohol, and vinegar, is really injurious to health. 87% of public health officials affirm that it is true chemical food preservatives are injurious to health, according to Wiley and the study. 95% of the surveyed public health officials deem it is not advisable to add preservatives of any matter to the food, and if it is able to be perfectly preserved without its usage. Preservatives are beneficial in their purpose in the food industry, but have been researched to cause hyperactivity and other harmful effects in humans and specific groups, according to a study. Sodium nitrate is used in meat product during cooking to prevent botulism, but during high heat, it reacts with proteins to produce carcinogenic N-nitroamines, and it can also cause anaphylaxis and severe itching. Sodium benzoate preservative food products are strictly prohibited for asthma patients. It forms carcinogenic benzene in low amounts, but still is considered a risk factor for those with cancer, for cancer and brain damage.
So now to finish with the conclusion. So the results clearly demonstrate that the use of citric acid and sodium benzoate does successfully prevent the growth of E. coli and salmonella on food. Now these results also help support the statement that food preservatives do effectively prevent the growth of food spoiling bacteria. And you may also notice that within our results that we didn't really have any results that really had the single, just using sodium benzoate or citric acid. They were used together, so obviously they have some type of synergistic effect together. However, do they work by themselves? Yes, they 100% do. However, together they obviously have a better effect than if they were by themselves. Even though they do work, they work better together. And then under our conclusions, we have our acknowledgments and also our references.